Hello and welcome to Science of Verity. In this episode we are looking at muscles, how they are formed and how they contract. There are three main types of muscle, all which perform a different function. Smooth muscle is found in the lining of the gut and in blood vessels. They perform unconscious contractions and have non-striated strands. Cardiac muscles are found exclusively in the heart and also have involuntary contractions. Skeletal muscle makes up the bulk of the vertebrate's body muscle and undergo conscious muscle contraction. They are striated and so have light and dark bands running the length of their fibers. The macroscopic structure of muscles occurs because the muscles are made from millions of tiny fibers known as myofibrils. These myofibrils are grouped into units which are bundled together because a group effort provides more powerful contraction, and the fibers are arranged into parallel lines as this gives the maximum amount of force. The cells are fused and share nuclei and a particular type of cytoplasm known as sarcoplasm. This contains a large amount of mitochondria and endoplasmic reticulum. Myofibrils are tiny fibers which make up a single muscle fibre. It is formed from two filaments which give it a striped appearance due to the way that they are arranged. Actin is a thin protein that is arranged in two strands, whereas myosin is thicker and has rod-shaped fibers combined with globular protein heads. Two further proteins are also present: tropomyosin, which is a fibrous protein that wraps around the actin, and troponin, which is a globular protein involved in muscle contraction. The microscopic structure of a muscle fibre zooms in on one of the myofibrils, looking at how the striations occur. The striping is due to different coloured bands. Isotropic bands are the light bands which are found. Light because the actin and myosin do not overlap. At the centre of this section there is a Z-line, which separates two sarcomeres. The darker colour is known as an anisotropic band, which has a slightly lighter colour in the centre known as the H-zone. It is darker because the actin and myosin do overlap. There are two types of muscle fibres: fast twitch and slow twitch. A fast twitch fibre is so called because it produces lots of short but powerful contractions over a short period of time. They have thicker filaments with a higher concentration of enzymes used in anaerobic respiration, as well as a ready supply of phosphocreatine, which is used to regenerate ATP from ADP and inorganic phosphate. Slow twitch fibres produce weaker contractions, which are less frequent over a longer period of time. They prevent the build-up of lactic acid due to the aerobic respiration which they are adapted for. They have a large amount of myoglobin for storing oxygen, as well as a rich supply of blood vessels for receiving oxygen and glucose. There is also a store of glycogen for metabolic energy. There is also a large number of mitochondria where the ATP is produced. The neuromuscular junction is the area where a motor neuron meets the muscle fibre. There are many of these junctions along the length of a muscle fibre, ensuring a single powerful contraction. Each neuron will act on a set of fibres, causing them to act as one unit. The number of motor units which receive the impulse dictate the amount of force exerted. A small number of motor units means that a little force is exerted. Sliding filament theory is a mechanism formed of cross bridges between the myosin and actin filaments. The I bands narrow and the Z lines move closer together, suggesting a movement, as well as the narrowing of the H-zone found in the anisotropic band. However, this remains the same size, discounting the theory that the filaments shorten. Myosin is formed from two proteins: one globular head which has two bulbous formations, and a long fibrous filament which is made of hundreds of molecules. Actin is a globular protein that forms long helical strands, where the two strands wind around each other. Acetylcholine is the neurotransmitter that crosses the neuromuscular junction. As the impulse is received, the fusion of vesicles with the presynaptic membrane occurs, and this is caused by an influx of calcium ions into the presynaptic knob. The acetylcholine is released across the synaptic cleft and diffuses to bind with receptors on the postsynaptic membrane. This causes sodium ions to enter the postsynaptic membrane as the sodium channels are stimulated. The acetylcholine is hydrolyzed by acetylcholinesterase, an enzyme, into its components, and is allowed to diffuse back across the synaptic cleft to the presynaptic knob where it is regenerated. Muscle contraction is a process that continues for as long as there is a nerve impulse which stimulates it. The contraction starts off when the action potential from the motor neuron travels down through the transverse tubules which branch through the sarcoplasm. From here, the potential stimulates the release of calcium ions into the sarcoplasm through the calcium channels. The calcium ions bind to the tropomyosin, freeing the binding sites on the actin which were previously blocked. An ADP molecule attaches to the myosin head, and this forms a cross bridge with the binding site on the actin. The myosin head changes its angles and pulls along the actin filament, releasing the ADP molecule. An ATP molecule attaches to the myosin head, and this detaches from the actin molecule. The calcium ions stimulate ATPase to hydrolyze the ATP, forming a molecule of ADP and one of inorganic phosphate. The energy released causes the myosin head to resume its original position. The ADP molecule means that the myosin head reattaches to the next actin molecule to continue the cycle. Muscle relaxation occurs when there is no more stimulation, and ATP is used to actively transport the calcium ions back to the endoplasmic reticulum. The reabsorption of calcium ions means that the tropomyosin repositions itself, blocking the actin binding sites. This means that the myosin heads cannot bind to the binding sites, so the contraction ceases. During a contraction, energy is supplied from the hydrolysis of ATP to power the movement of the myosin heads, disbanding them from actin and resuming their positions, as well as the reabsorption of calcium ions in the endoplasmic reticulum. Muscles have a great demand for ATP, which is generated from both aerobic and anaerobic respiration. Active muscles, such as slow-twitch muscle fibers, use aerobic respiration, which requires oxygen to generate ATP, helping to prevent a build-up of lactic acid. This is unlike very active muscles, such as fast-twitch fibers, which tend to produce ATP through anaerobic respiration using phosphocreatine. Phosphocreatine is stored in the muscles in readiness and is used to regenerate ATP by providing a phosphate group to bind with the ADP molecule. The store is built back up while the muscle is at rest, allowing a reserve supply to form. So, to sum up, there are three types of muscles: skeletal, which is conscious contraction, and cardiac and smooth, which have involuntary contractions. The fibers are formed from bundles of tiny fibers called myofibrils, which are then grouped into slightly larger groups. The myofibrils are formed from two proteins, myosin and actin. There are two types of fiber known as fast-twitch fibers, which use anaerobic respiration to regenerate ATP, and slow-twitch fibers, which use aerobic respiration to produce ATP. The neuromuscular junction is the spot where the neuro meets the muscle fiber. Many of these are found along the length of one myofibril. The sliding filament theory is recognized as how the actin and myosin interact with each other. The myosin head attaches to the binding sites on the actin after the release of calcium ions, which free the binding sites by binding to the tropomyosin wrapped around the actin. The calcium ions also encourage ATPase to hydrolyze ATP, and the diffusion of calcium and lack of stimulation cause a muscle to relax. ATP is used to provide energy and is produced by both aerobic and anaerobic means. Thanks for watching. Science with Verity.